Susan asked me to talk about drug development for Alzheimer's disease, and I'm very happy to do that. It's one of the central activities that we're engaged in here at the LouRuvo Center. So drug development just means trying to find new, better treatments for people. But we call it drug development. That's kind of the code word for what it is that we do. And the reason we do it is because our treatments are inadequate, right? The things that we have, the drugs that we have to offer to patients are not enough. They do not arrest the disease, and they have a modest amount of improvement at best. So we need better drugs, and so we engage in drug development. Now, it's also true of every neurological disease. If you have epilepsy, we are not able to control the seizures completely. If you have a brain tumor, we're not able to treat it as well as we want. If you have Parkinson's disease, you still will have Parkinson's symptoms when you're optimally medicated. So we need new treatment development for every neurological disease. So I'm going to take you through how we do this for Alzheimer's disease, but you'll see right away that this is applicable to many types of neurological disorders. I would ordinarily take questions as I go along, but Susan does not allow that. Because she says when we're filming, then it takes it sort of in many directions, and she wants all the questions at the end. So this is a memory test. Save your questions till the end, and then we'll review them. I also thought we were going to have the big screens today, so I'll help you read these slides, but they're a little bit smaller than I would want. So here's how we get to a new drug. We do basic science. This is in the laboratory. We take basically some cells and figure out whether they're abnormal, and then can we make them more normal with some chemical. When that looks good enough, we do preclinical research. This is mainly on animals. We have animals that are genetically engineered to resemble Alzheimer's disease. And then we would give the drug to the genetically engineered animal and see if it helps them. And if it does, then we would go into clinical trials. And clinical trials is what we do here at the LouRuvo Center for Brain Health to see whether these drugs help patients. So this is basically this process that we're engaged in for every drug. And the FDA that ultimately approves drugs will only accept medications that have been tested in clinical trials. So every drug for every disease, whether it be Alzheimer's disease or the fungal infection in your big toenail, it has to go through this same process, every single disease. So clinical trials are the only way to develop new therapies for any kind of condition. And I'm just going to take you through this process so that you understand it a little bit more. See what we're doing here at the LouRuvo Center. So how would we do this first step, this basic science? Well, very often it starts by studying the brain of a patient who has died of the disease. So it would start with a patient who has gone all the way through the course, and we look at the brain and find the pathology. And in this case of Alzheimer's disease, there are tangles, neurofibrillary tangles, and there are plaques in the brain. And Alzheimer described this in 1907. That's why we gave it his name. So he looked into the brain. He had this brand new technology, which was a new kind of stain, and the stain allowed us to see these plaques and tangles. Later, it was quoted as saying, "The gains in the brain are mainly in the stain. That's how we think of Alzheimer's work. So then we see, well, now I see that we have plaques and tangles. Are some of these druggable? That means, could we do something to plaques and tangles to prevent them? Is this a druggable target? And then we would start with a bunch of chemicals to see whether they reduce these plaques and tangles. In cells, for example, we would test them in cells first. So that's this basic science part. So it really starts with a patient, trying to understand what's going on in the brain of the patient, and how does that lead to this disease? Can we interrupt it? So where do drugs come from? Right? Now I understand my process. I got plaques and tangles. Where am I going to look? Well, nature. Nature still accounts for 40% of all the drugs that are ever developed. They come from flowers, they come from bark, they come from roots, berries, and there's all sorts of drugs. That's why we want to preserve actually the diversity, the biodiversity. Let's say South America. We want to preserve those rainforests because there's a whole bunch of drugs down there that we haven't discovered yet. So it's odd. You know, you start off with Alzheimer's disease, and now you're talking about preserving rainforests. But it's all connected. So nature is an important part of this. Traditional medications are an important part of this. Chinese are a good example. Chinese traditional medicine. There are about, 40,000 compounds or something that the Chinese have studied over 2,000 years. And we can see that some of them actually help Alzheimer patients. There's a drug, for example, called Huperzine that the Chinese have known about for thousands of years. And it is exactly the same chemical that we call Aricept, and is the most commonly used drug for the treatment of Alzheimer's disease. And the Chinese have known about that for centuries. So it's an example of traditional medications. Huperzine is still actually in clinical trials right now. What big pharmaceutical companies have are libraries of agents. So a company like Pfizer, I think they have a library of about 3 million drugs. So when they have a new target, like an amyloid plaque, they can figure out a cell that produces the plaque, and then they could add up to 3 million drugs in that little cell to see whether or in 3 million cells, one cell per drug, to see whether their library has a promising agent for the treatment of Alzheimer's disease. One of the things that we do a lot of here, because we can get drugs to patients faster, is testing repurposed agents. So you know that we've been testing a drug called bexarotene. This was a drug which was approved several years ago for cancer, a special kind of skin cancer that elderly people get. And because of the properties of this drug, an investigator in Cleveland gave it to these genetically engineered mice that get the changes of Alzheimer's disease. And it produced a 70% reduction in the protein that causes Alzheimer's disease in 6 days. 6 days. So, I thought to myself, we ought to see if that works in people. Brilliant insight, right? Let's see if that works in people. We've cured a lot of mice of Alzheimer's disease, by the way. We're just hoping to get it a little further up the chain. So that's an example of a repurposed agent. We just finished that trial last week. We're just waiting for the results of the brain scans to be read. And I should know in the next month whether that drug had a similar effect in humans as it did in the animals. But my point is that there are a lot of repurposed drugs. Another drug we're testing has been shown to work and be useful in Parkinson's disease, but it's never been tested in Alzheimer's disease. So that's a repurposed drug. The benefit of a repurposed drug is we already know how to make it. We already know what its side effects are. We already have distribution channels out to get it to a lot of patients. So there's a whole bunch of the groundwork of drugs that's already done for repurposed agents. So we like these drugs. Computers, just like computers are involved in every aspect of our life, so they are involved in drug design. So you can actually just design a drug on the computer, and then you can test it in the computer. Does it fit into the receptor site? Does it look like drugs that cause a lot of side effects, or does it look like a drug that has few side effects? So you can actually test the drug in the computer, and you eliminate thousands of compounds that for some reason or other would be unacceptable. And then you just synthesize the one that looks really promising, give that to a person. So you can do a lot of drug design in the computer, called in silico design. And then one other way is to just take a drug. Let's take a drug like risperidone is a commonly used tranquilizer. And it's actually metabolized into another drug, and that drug is also a tranquilizer. So one way to simplify things is just to take that metabolite and make it a drug. Rather than taking risperidone, you're actually taking two drugs. You're taking the risperidone and you're taking the metabolite. So introduces a lot of complications. So my point is here, we've got a drug target, plaques and tangles. Now we have to develop some candidates that might be really new treatments for Alzheimer's disease. How would we go about it? And this is how we go about it. We figure out all these different ways, and one of the most common ways we use is the repurposed agents. And when we work with pharmaceutical companies, we're mainly working with drugs that have come out of these libraries. So these are the two most common ways of drugs that we use here at the LouRuvo Center for Brain Health. So now we've finished our little basic science here. Now we're going to go on to preclinical research. And the preclinical research is usually done in animals. We do everything we can to minimize the use of animals. We also ensure the safety of animals. But we do have to see whether the drug is toxic before we give it to a human being. And rats are commonly used. There's some jokes about rats and attorneys that I'm not going to go into right now. And dogs are commonly used for the cardiac effects. Dogs are particularly prone to develop cardiac effects of medications. So if you give a drug to a dog, take its EKG, you can see whether that drug might affect a human being's heart. So we look for rats and dogs, and the FDA requires that a drug be tested in at least 2 species. So rats and dogs. And so this is not something that a pharmaceutical company can choose not to do. They have to do this by law. And then, of course, we look for efficacy. That is, does the drug seem to make some aspect of Alzheimer's disease better? And for this, we use these genetically engineered mice. They're really terrific, although so far they have not actually predicted a successful drug, but they at least show us that the mechanism is working. And we use monkeys because monkeys are the closest things to humans. So if we give a drug to a monkey and then measure the spinal fluid level of the drug, we can see whether it's actually getting into the brain area. Well, that's pretty important. Before you give it to a human being, you need to see whether it's actually getting into the brain. So this is usually what's done, and they're done, one group of scientists are specialists in toxicity, and one group of scientists are specialists in efficacy, and so they're taking these down parallel so that at the end, if we have a safe drug that seems to be working, then we can take it the next step and take it into human clinical trials. This is a basic science is a couple of years, preclinical research is a couple of years, and most of the time of a drug's lifespan is spent in the clinical trial. So I made this point before, that clinical trials, this last phase, are the only way to get a new drug approved. So if I want to advertise, make sure that as many patients get this on my drug as I can, it has to be approved by the FDA. And the FDA is involved in this process from the very beginning. So the FDA would look at your basic science and say, yeah, we think this is pretty promising. They would look at your preclinical science and say, yeah, the animals are safe enough and looks pretty good. And then they keep consulting all the way through the clinical trial process. So it's not like the FDA is some sort of ultimate judger out there, that you bring all your data and you hope for the best. It's not like that. The FDA is in constant contact with the companies and the investigators so that when you get to the FDA final submission, you've actually met with them many times, and there's no surprises involved. So when we get to this clinical trial, this part of drug development, we go through phase 1. I'm going to tell you all about that. Phase 2, phase 3, and at that point, we're prepared to present our drug to the FDA. So there are 3 human phases of drug development. Here at the LouRuvo Center, we do phase 2 and phase 3. In Cleveland, they do have a so-called phase 1 unit. And phase 1 usually involves normal people, okay? And this is the first time that a human being has been given this drug, right? Risky business. So, you've tested in animals, but, I'm not too comfortable with this, just because a drug has not made a rat sick doesn't make me feel real confident about that drug, but somebody has to do it. I had an interesting experience. I was in Washington, D.C., and cab driver was taking me from one place to another, and he said, "What do you do?" And I said, "Oh, I'm an Alzheimer guy." And he said, "Oh, I do these phase 1 trials." And it turned out he was a volunteer for the phase 1 trials. And so part of the time he's a taxi driver, and part of the time he's in the phase 1 unit. And he would go in and volunteer to take a drug. The person is given first a very low dose. Everything goes well, and they're given a higher dose, okay? Everything goes well, up a little bit more, until you finally find something called the maximum tolerated dose. And that might be either because they started to get sick, or because they started to get sleepy, or maybe their pulse went too high, or their blood pressure went down. So you'd figure out how much of the drug can be tolerated, and is it safe? Having any effects on the heart or the liver or anything. You'd also measure how does the human body metabolize the drug, because until now you only know how the animals are metabolizing the drug. There's a lot of difference between a rat and a human, right? Despite how we feel about some people, there are still a lot of differences here. So we don't do this at the LouRuvo Center, but there is a phase 1 unit that we can use at the Cleveland Clinic on the main campus. So that's phase 1. It's all normal people, and they have vast databases of people who will volunteer. We usually, for an Alzheimer drug, we test it in normal young people, and then we would have one group of normal older people. So we'd get some sense of, well, do older people metabolize this drug very differently than younger people do? Because we know that most of our Alzheimer patients are going to be older. And therefore, we want to have some sense of how the drug is handled in a normal elderly person before we give it to an elderly person who has Alzheimer's disease. So we would test this usually in young normal people and in older normal people. Now we go to phase 2. This answers the critical question, Does the drug work? So now you must give it to the population of patients for whom the drug is developed, like Alzheimer's disease. So you want to know two things. One is, does it work? Does it make people better? And what is the dose? And dose is kind of tricky because you don't want to give people doses that are so low that they're irrelevant, and you don't want to give them so high that they're toxic. And you're guessing, based on how the animals did, what dose you should take into humans. And if you haven't done very much of the decision work, you might have 4 doses. Well, to test 4 doses in a lot of people is a lot of work. You'd rather test 2 doses so that you can get fewer people into the trials. So dosing is a very tricky part of this, and there are whole scientists whose whole specialty in the world is to figure out how to do the dosing. So we do phase 2 studies. So this is in the middle of the development. So bexarotene, the cancer drug I just described, that's a phase 2 study. Rasagiline, that's the Parkinson's disease repurposed drug I just described, that's a phase 2 study. And then one of the antibodies that we've tested here to see whether we can get the immune system to work better in Alzheimer's disease is crenezumab. Any drug that ends in mab means it's an antibody. And so I just reported on this drug 2 weeks ago at the international meeting in Copenhagen because we were the lead site and I was the lead investigator in the world on this crenezumab trial. And it looks like it worked in mild patients. Did not work across the whole population, but when we looked at the mild patients, we got a very consistent signal, which might mean, and now we'll test this specifically, that if we give the drug to early patients who have just entered having their first symptoms, we'd be able to largely arrest this disease. So that would be fantastic. One of the tests had a 68% difference between the drug and the placebo. So it was really very exciting. So this drug is owned by a company called Genentech up in San Francisco. And so now they will decide whether or not are those data enough that they're going to invest in the clinical trials to test it in mild patients. It's a serious decision because your average clinical trial cost about $60 million. $60 million. So, and this would not be an average. It probably would be more expensive than that. So companies spend a lot of time trying to decide whether a drug is good enough in phase 2 that it can go on to phase 3 because it's going to be very, very expensive. So let's talk about phase 3. Okay, now we're really happy. Our drug looks good in phase 2. It's safe. And now we want to see, can we actually make it available to patients worldwide by getting FDA approval? So these are usually large trials. We usually have between 1,000 and 2,000 patients in these trials. Huge. Many clinical trial sites are involved because in general, we only get about 10 patients per site. Isn't that amazing? Here's the world's most common disease, and we can only get 10 patients per site. It's because there are lots of inclusions and exclusions in these trials. So you can't be on this drug or that drug. You have to be younger than this but older than that. You have to have a mini mental state or general memory problem between X and Y. And by the time you get all of that shaped, you get down actually to quite a few patients, just a few patients. So we would be doing really great if we get 10 patients into a trial from our site. And in general, we're running, maybe 10 trials at a time. So we have roughly 100 patients in trials at any one time. But it would be distributed across a whole set of different trials. This produces a huge amount of variability, because you can see that you have to have at least 100 sites and maybe 200 sites if you need 2,000 patients and you're only getting 10 per site. Well, we're a really good site here. And I see our site people here. We're really good. We really know what we do and we really care about the data. But there are a lot of sites that are not very good. And they just kind of work patients through, and they don't really care very much about how they test, and they're just, because there is a payment involved from the pharmaceutical company. When we give them data, you get a payment for those data. There are companies that are just motivated to get the payment rather than to get the best possible data. So this is a huge problem in our field. One, I'm also very concerned that when you have 100 sites, you almost always have to have them around the world. And you can imagine, deciding, well, here's the LouRuvo Center for Brain Health, and here's Peking Union Medical Center, and we're trying to test the same drug with the same group of tests. Well, there's quite a lot of difference between those two sites. So we're trying to understand how to make these data ever better. So these phase 2-3 trials are the FDA preparation, and we do a lot of phase 3 trials. An example here is solanezumab, right? You've just learned that mAb means antibody. So this is another antibody. This is from Lilly. Looked pretty good in phase 2, and so they're advancing it to phase 3. So that's an example of the phase 3 studies that we're doing here at the LouRuvo Center. Well, placebos. Placebos are the best and the worst drugs, right? They have all the effects of a drug. And they also have, in fact, they have all the effects of all drugs. Because when you give a placebo, you almost always have a placebo response, whether it's Alzheimer's or Parkinson's or epilepsy. You have a little bit of a placebo response. So they have all the effects of all the drugs. They also have all the side effects of all the drugs. And when you give a placebo, you will always have some people who have diarrhea over the next 2 weeks, or they have some vomiting over the next 2 weeks, or they have, whatever. So there's always side effects associated with placebos. But that's why they're so important, is because if the effect is greater than a placebo, you can believe it. And if the side effects are more than a placebo, you have to worry about them. If they're at the same as a placebo, you don't have to worry about them. So the placebos are really informative. Moreover, they are required by the FDA. Because the FDA says we don't believe any drug that hasn't been compared with a placebo, because we know that placebos can make you better and placebos can make you worse. So your drug has to be compared to the placebo, or we will not accept these data. Now, one way we get around this and to help our patients is that most trials have a period of guaranteed treatment after the end of the placebo period. So let's say we're doing a 12-month trial. In the first 12 months, the patient and we would not know whether they were on drug or placebo. That's all known only to the pharmacist, somewhere in Cleveland in our case. We can find out if somebody develops big-time toxicity. We break the blind and find out whether they were on drug or placebo. Short of that, we would not. After that year period where we're comparing drug and placebo, then we would typically put everybody on the drug for an additional 12 months or 18 months, however, until we figured out whether the drug was working. If there's no drug-placebo difference, then we would say to the patient, there's no sense of you being on this drug, we stop it. On the other hand, if there is a drug-placebo difference, then we would work with that patient to try to continue the drug in that patient. So the placebos are very important. Now, one of the things that we have done also in recent trials, which I think is important, is, let's say Aricept is the most commonly used drug for Alzheimer's disease. Well, we put everybody in the trial on Aricept. And then half of those patients get the new drug, and half of those patients get placebo over the next few months. But everybody gets the accepted standard of care, so that the trial should never be in a situation of depriving people of the best standard of care. The trial should always be about taking it up to the next step of what is the next best level of care. So we don't have really true placebo groups. We have Aricept groups that are being compared to the new drug. Well, how do we know if a drug's working? This is critical because most drugs have relatively small effects. So it's not as easy as one would think. Of course, if a drug had a big effect, and you give it to mom, and the next day she gets up and she's redirecting your investment portfolio, you say, wow, this drug is working, right? Right? No question about it. But in most cases, it's a lot more subtle than that. So how do we decide? Well, we do clinical measures to see if the patient is functioning and thinking better. We do tests. Say, well, here are 10 words. How many of these 10 words can you remember before the drug starts and after the drug and compared to placebo? So that's one way we do it. And now we have these great biological measures, mostly brain scans, but we also measure changes in spinal fluid in some trials. Spinal fluids involves having a spinal tap. Right, so you have to have a spinal tap. We remove the fluid and take it out of the back here and analyze the fluid and see whether the drug had any effect on the fluid, because the plaques and tangles show up in the fluid. So we can see whether we're having any effect on plaques and tangles. But oddly, many people do not want to have a spinal tap. So we use the brain scans instead, and only a few trials are we using the spinal tap. In Europe, Europeans seem to like spinal taps much more than we do. And so there's many more spinal fluid studies in Europe than there is in the US. So what about the kind of clinical measures? Well, we'd have a global rating. They're called global ratings, and they're basically based on the caregiver saying, yeah, the patient is better, or no, the patient is worse, or there's no change. Then we test memory and cognition. We query the caregiver about activities of daily living. Is he able to dress? Is he able to feed himself? Is he able to engage in conversation? Is he able to prepare a meal? Could he ever prepare a meal? These are all the things of activities of daily living. And then behavior is the fourth critical element, because we know that in Alzheimer's disease, people get depressed, or they get anxious, or sometimes they have hallucinations. And so measuring them and see whether they're less in the treatment group compared to the placebo group is a very, very important aspect of trials. And it's actually how I got into trials, because I invented the scale that is most commonly used to measure behavior in clinical trials. It's available in 40 languages, so it's widely, widely used. And then sometimes we measure quality of life, sometimes we measure how the caregiver is doing, and sometimes we measure resources and costs of care. Hoping to discover, of course, that patients who are on effective treatment require fewer resources and have less costs. That's what we'd like to discover. The caregiver, very important. You would say, well, shouldn't we be measuring quality of life in every trial? And one of our problems right now is we have really lousy quality of life measures. So they tend to say, well, how's your financial situation? How's your relationship with the patient? How's the relationship with the rest of your family? And how's the person's function? Well, of those four huge areas, the drug could only affect one, because there's no drug that's going to make you richer. There's no drug that's going to make you like your spouse more. There's no drug that's going to make your family work more closely with you. So our quality of life measures don't work very well in clinical trials because they measure the wrong things. And that's one of the things that I'm very involved in, is figuring out, can we get quality of life measures that might reasonably be moved by a pharmaceutical intervention. Biological measures, this has become, again, so exciting for us. We measure MRI. Is the brain shrinking less? Because in the course of Alzheimer's disease, the brain shrinks down. So is it shrinking less on our new treatment? That's critical. We use PET, that's positron emission tomography, and it's a way of looking at all kinds of cool elements like metabolism. We inject the person with radioactive sugar. The brain uses the sugar, and the drug might make the brain use the sugar better. And then we could compare it to the placebo and can see whether there was more metabolism in the treated patient compared to the placebo patient. That would be a key finding with PET therapy. We do amyloid imaging. Is amyloid removed? Remember, amyloid is the protein of the plaque. And we're trying to get rid of those plaques. That's one of the things we'd like to do. So is the plaque being removed by some of our therapies? We did amyloid imaging before and after treating people with bexarotene. And that's what we're going to find out very soon, is whether or not we actually reduce the amyloid in the brain. And then, as I say, we sometimes do CSF and a lumbar puncture. But this is the kind of thing. Here's amyloid imaging. Here was an antibody. Here's the treatment group before and after 78 weeks of treatment. Here's the placebo group before and after 78 weeks of being on a placebo. And I think you can see that there's less of this red in the treatment group and more of this red in the placebo group, because the placebo group continued to get more plaques in the brain, and the treatment group had fewer plaques in the brain. So now we compare the drug and the placebo after the treatment period, and we can see that the drug group was doing much better than the placebo group. So that's the kind of the way that we would use amyloid imaging to help us figure out whether a drug was working. For the most part, it's easier to see an effect with brain scanning than it is on memory testing, because we all have good days and bad days. We have days where we're just hitting on, and those names are coming to us just like this, and there's no hesitation. And we have days where we had a bad night's sleep before, or we had a little bit too much Pinot Noir, or something, and the result was that you're just not hitting on all cylinders. And that'll be reflected in your memory testing, but it will not affect your brain scan. So there's a lot more variability in human testing than there is in the biological markers like scanning that we use. So let's finish this up by talking about kind of the end and about pharmaceutical companies. It's important to know that pharmaceutical companies produce 97% of all drugs. So the government doesn't, the post office doesn't, right? Academic medical centers don't, right? They're all done by drugs. They're all done by drug companies. So people have a lot of different responses to drug companies, but I'll tell you, without them, we are down to 3% of possible drugs. That's not where we want to be. The costs are roughly $1 billion per successful drug. So those three phases I showed you, from basic science to preclinical to clinical trials, if you add that all up, it's about $1 billion. A lot of money. Failed drugs are costly and they have no return, right? And most drugs fail. So the successful drug has to pay for all the failed drugs. So it's a very complicated economy that the pharmaceutical companies are involved in. We just wrote a paper that showed specifically that over the last decade, 99% of all Alzheimer drugs failed. Only one succeeded, memantine, in the past decade. You know, that's a heck of a business model when 99% of your products fail. Nevertheless, that's what we're in. And I'm working very hard with my group here and other groups all around the country to make sure that this 99% figure does not continue to characterize our field. But one way of thinking about this is pharmaceutical companies and the government. All right, so I realize these are small, but if we look at the National Institutes of Health, their total budget for research is about $30 billion, $29.2 billion, okay? That's all of NIH. That's National Institute of Mental Health, neurological disease, aging, cardiovascular disease, cancer, all of NIH, $30 billion. If we look at the pharmaceutical companies, their annual research budget is about $50 billion. So pharmaceutical companies are investing almost twice as much in research as the federal government is. So, important to think about that. By and large, the government invests in basic research. How do these cells work? How can we create new mice? We need better mice. They invest some in translational research and a little bit in clinical research like clinical trials. Pharmaceutical companies do the opposite. They invest quite little in basic research. That's not their basic business. They invest a lot in translational research like new biomarker development, and they spend most of their money in clinical trials preparing for the FDA presentation. So these are two complementary systems in our country, and one without the other would never work. We have to have both of these systems because ultimately these clinical trial drugs come out of the basic research funded by the government. So these two are interactive, mutually intertwined, but a lot of people don't realize that pharmaceutical companies are investing almost twice as much as the government in overall biomedical research. So it's important to think about that. Challenges, as I say, most drugs fail. Slow recruitment. Okay, I'm emphasizing that, slow, because it's hard to get enough patients into clinical trials. You would think, again, with a common disease, it wouldn't be, but it is, because most people, I think something like 7% of all people in the world, all people in the United States will be in a clinical trial. But 100% will have an illness, right? So 93% of people with an illness are not participating in the clinical trial. Patient and caregiver burden, of course, it's a pain in the neck to come over here to the LouRuvo Center and have your testing done and have your memory tested again and again. We appreciate that. Site variability, I emphasized that already. The effects of mice often do not predict the effects in humans. This is a basic problem we're trying to solve. And then that's true of both side effects and efficacy. So I'll just summarize. This is a really complex operation. We're an important part of it in Cleveland Clinic and the LouRuvo Center. Clinical trials are absolutely critical. It's the only way to get a new drug. We're one of the major sites here. It takes a long time, several years, to get a drug out of the laboratory and to the FDA. And participant volunteers, so inviting our patients, are critical to the success of this whole enterprise. In a way, the patients hold the success in their hands because no clinical trial can be done without patients. So smart as the doctors might be, smart as the scientists might be, they have no power to advance a new drug without the alliance of the patients and the families. So that's why we enjoy it. We enjoy doing clinical trials, is because it creates a unique alliance between us and our patients as we move towards new therapies for Alzheimer's disease. Oh, I'll stop there and take any questions that might have occurred to you. Yes, sir.
Is there a placebo effect in any other species besides humans?
What an interesting question. Is there a placebo effect in any other species? Wow. Not that I'm aware of. But that's such an interesting question. Like, for example, would monkeys or chimpanzees or some very high species have a placebo effect? They would have to have expectations of therapy. And I don't think they have expectations of treatment. But it would be interesting to think about that more.
So it would lead one to try to understand how veterinary drugs are how their trials work, What are they based on, too?
Right. Well, so there are no, in veterinary medicine, there's no Alzheimer's disease. We actually have to create it in mice. But liver disease is common, and you would actually do a drug placebo trial in horses to see whether your medication was an equine liver treatment. You have to do it. Yes, sir.
What drives the high cost of clinical trials? Is there one or two particular factors that drive most of it?
So the question is, what drives the high cost of clinical trials? I think it's mainly, well, there are two aspects. Personnel on one hand, because it takes a lot of time to administer all these tests, collect all the data, analyze all the data. And the other is the imaging. The brain imaging is actually quite expensive when you're doing it on thousands of patients, because an image might cost somewhere between $1,500 and $3,000, and you're doing that maybe multiple times in the course of the clinical trial on thousands of patients. So the brain imaging is one of the things that drives up the Alzheimer costs very greatly. There's also an uncomfortable amount of inflation, because there's a middleman between the pharmaceutical company and the site, and they're called contract research organizations. And they are responsible for organizing all of the sites that are participating and collecting the data and giving it to the sponsor. And they charge exorbitant fees. And we think that's a place where we can work. It's one of the things we're calling attention to right now. Yeah. Another question, yes, sir?
Yes. Drug interactions, do you measure what effects people are getting from, let's say, taking statins or blood pressure medication while you're doing your trials? And are you using any pharmacogenetics to screen your pre-trial candidates or looking for effects afterwards?
Yeah, great questions. You're obviously knowledgeable about this process. So drug-drug interactions, we know how a statin is metabolized, and we would know how the experimental agent is metabolized. That would be developed in some of the preclinical work. And then we would be able to know whether we should test them together because an interaction is likely. So we usually are forced to do maybe 4 or 5 drug interaction trials, like with a blood thinner, with a pain medication, with an anti-infective medication. But of course, there are thousands of drugs, so you can't test a drug with every drug. But you can test them with the ones where there's most likely to be an interaction that would be injurious to the patient. So we do usually have some drug-drug interaction, and we have knowledge of likely drug interactions.
Do you do that in phase 1?
We do that we do it in phase 1 and at the preclinical level. The pharmacokinetics are characterized preclinically and then continued in phase 1. In terms of pharmacogenetics, this is, do your genes somehow predict your response to drugs? And we know that that's very important. We only test for one routinely at this point, which is the so-called ApoE4 gene. And it makes it very likely, it increases the likelihood that someone will get Alzheimer's disease if they're carrying this gene. But we also think that it is likely to affect the activity of many of our drugs. So then in our analysis phase, we'd say, okay, we're going to test first we're going to see what happened in the whole population. And then we would say, now we're going to look at the statistics just in the people who have the gene, and now we're going to look just at the statistics in the people who don't have the gene. So we do a basic level of pharmacogenetics, but I think we need much, much more of that. And I think the genetics can really be helpful in this 99% failure rate. Because if we could choose the patients better, so that we know, well, this patient has this gene and is most likely to respond to this drug, then we could, then we have much better chance of success. Right now, we just give drugs to everybody, and we're as different as our faces are. That's how different our genes are. Yes?
Where do vitamins, minerals, and natural herbs come in on this? Since you said in the very beginning, most of the chemistry that we're taking comes from, for instance, the Amazon. Has to be chemicalized and processed and preservatives in them, et cetera, when what happened to the flower in the beginning?
Yeah, good question. So the question is, what about herbs and vitamins and minerals, and how does all that fit in here? Well, of course, they have chemical effects. So the Euperzine that I mentioned as a Chinese traditional medicine, that's an herb. So that's an example of an herb which has been tested through the usual mechanisms and shown to be effective in relieving some of the memory problems of Alzheimer's disease. If we look at vitamins, there are actually 2 positive trials of vitamin E in Alzheimer's disease. And both of them, vitamin E reduces the injury to the tissue produced by Alzheimer's disease. It's an antioxidant, and these are oxidative injuries. So why don't we use it well, use it more? It's because the effective dose is 2,000 international units. This is the same dose which is known to cause cardiac effects, adverse cardiac effects. So we are afraid to routinely prescribe the dose of vitamin E that we think is required to have this beneficial effect on the brain, because we don't want to trade a brain problem for a heart problem, right? But there's clearly a way forward here, and there is a green tea extract, which is a very powerful antioxidant and is in advanced phase of clinical trials right now. So there are examples of herbs and vitamins, I'm not sure about minerals, where you look at their chemical properties, you see if they're doing what you want them to do, you test them in a routine way, and they could become treatments for Alzheimer's disease.
FDA wants to take a lot of these vitamins and minerals off the shelf. Is that why? Because of self-dosing and overdosing?
So the question is, what about taking a lot of the minerals and vitamins off the shelf? I don't know exactly what the increased scrutiny is about. I know that people have made an awful lot of claims for certain vitamins and minerals that were not well substantiated. And I think the FDA wants the truth in labeling, so that you actually know, if you're taking super brain power, does it actually have any help for your brain? It would be a good thing to know. So I think they're working on that. Yes, sir. Then I'll come over to you.
Proteomics, see what gene expression you get after giving the drug, and maybe with dosing and all of that.
So the question is, what about proteomics? I love these words. This is fantastic. So a gene becomes first RNA, and then it becomes a protein, and then it becomes something like a vitamin or mineral. So you can look at any of those phases, and when we look at proteins, we call it proteomics. And we are just starting that. I just talked with a company this week, and we're in the rasagiline trial. We're going to introduce multi-omics. So we're going to do all 4 of the levels. We're going to go do genomics, transcriptomics, proteomics, metabolomics to get a multi-omic picture of the drug versus the placebo. So we're really stepping that up. I think there's going to be a lot of answers there, but of course the information processing demands of that kind of testing are very high. And that's another problem we need to solve. Yes?
Is the LouRuvo Center involved at all in the A4 study with the pre-symptomatic patients that have some kind of familial or genetic indicators of Alzheimer's?
Yeah, so this is a great question. Again, what an informed audience we have. So A4 is a federally sponsored trial where we're testing, again, solanezumab, actually the drug that we're testing in another trial here, but we're giving it to people who are normal, cognitively normal, but we have to know from their scan that they have amyloid in the brain, this protein in the brain. And then we give them the drug, and we determine whether or not fewer people who get the drug actually go on to develop Alzheimer's disease. How great is this, right? We're preventing Alzheimer's disease. Fantastic. Love these trials. So yes, we're doing that. We're recruiting right now, and we're just, we're not quite recruiting because it has to be approved by our internal review board, and they're just going back and forth right now, but we're almost ready for that.
And how would you be selecting patients for that? Because I have a familial history.
Yep.
And I would be very interested in the genetic testing. My father has the E4 gene.
So we would, you're too young.
Okay.
Congratulations, you are too young. Because we need people who are old enough that they would be in their Alzheimer risk years in the course of the trial. So that's usually people, let's see, my trial gang left. Oh, there's Samuel. Samuel, can you help me? What's the age range that we're wanting for E4?
63 to 85.
Okay.
Okay. I didn't hear that, but apparently other people did. Okay, great. So then, and usually we take a family history, and if there are more of these risk factors, then you're more likely to have amyloid in the brain, and therefore the scan would be more likely to be positive.
There's an early onset history too. My father started having issues at 60.
Right. Most people who have the genetic influence begin the disease earlier in life. Yeah. Yes, ma'am. I mean, this is going to be the last question.
You mentioned the tremendous cost to develop a drug. I was just curious, from the very beginning, to when the drug is actually approved by FDA, about how long is this normally taking? I know it would vary, but I was just curious.
Yeah, good question. So the question is, how long does it take to develop a drug? And on average, it's 13 years from the time that a compound is first characterized to the time that it would be presented to the FDA. So 13 years. So one of the things that this means, of course, it takes about a decade for a drug to be fully screened, get through the animals, and get into humans, where we're testing them here at the LouRuvo Center. That means the drugs that we're testing now are actually based on the science of 10 years ago, right? So, I mean, there's just some built-in problems with this system that we can't overcome, because you can't skip the steps in between, and yet all of them take time. So these are all the reasons that we have a high failure rate, but they're all addressable, and I don't want you to go away from this lecture with kind of a hopeless feeling, because this is complicated, but a lot of effort is being put into this from the government, from the FDA, from many other sources to try to make this a much more efficient process than it currently is, and I'm convinced it's going to happen. So thank you for coming today. Susan. Oh, yes. Thank you. Thank you to our group. They have a question? Okay.
Yes.
Sorry.
There was one question. She wanted to know if there's a typical age where people develop Alzheimer's.
Yes, 73.
Oh, great.
That's the average age. If you take all the patients in the world, of course it becomes more and more common as people get older. But if you took the whole population of Alzheimer patients and asked when it first started, it'd be 73.
And I have one other question. Does phase 1 patients who are normal, do they get paid stipends to participate in phase 1 trials?
Yes, so the phase 1 participants do get stipends. That's why they do it. So there is a payment for that. I don't know how much it is, by the way. It would have to be pretty high for me.
Probably depends on what the drug is supposed to
Yeah,
affect.
might be, yes.
Yeah.
So please join me in thanking Dr.